Hello friends, in this video I will tell you how coronavirus test is done, how we detect COVID-19 in human body. So first step involves collecting sample from patient body. It can be collected from throat or from blood. After collecting samples, extraction of genetic material of virus is done. The extraction of RNA is done by centrifuging sample along with some enzyme and chemicals. After extraction of RNA, this RNA is converted to DNA by using reverse transcription. After converting RNA into DNA, we need amplify it so we can detect easily because it is in very small amount. So this DNA goes into PCR machine which amplify this DNA by using polymer chain reaction. The fluorescent dye is also added which emits light in the presence of DNA. The positive and negative of test is determined on the basis of light emitted through the fluorescent dye. If the patient is not suffered from corona, then RNA is not present and hence DNA is also not present. So there is no light emitting. By this process, we determine the patient is suffered from corona or not.